I am Adam Davidovich. I'm a PhD candidate in the Feinberg Lab at Johns Hopkins University, and today I'll be presenting my work, Long-Read Sequencing Reveals Diverse Patterns of Epigenetic Inheritance in Mice. So it's been well established that DNA methylation and other forms of epigenetic information exist at the nexus of genotype in the environment, insofar as they are capable of being influenced by both and inherited in a multi-generational manner. Now, as such, the epigenome can be used to help explain the multitude of common complex diseases and phenotypes which exhibit high degrees of heritability but paradoxically report minimal genetic influence through genome-wide association studies. These include neuropsychiatric disorders such as schizophrenia and bipolar disorder, metabolic syndrome, and even autoimmune disorders such as rheumatoid arthritis. However, in order to study the epigenome as a source of this potential missing disease heritability, it is critical to first be able to understand the complex relationship between the genome, epigenome, transcriptome, and phenome, and then to further understand how this information is transmitted intergenerationally. So in order to begin studying this intergenerational regulatory dynamic, we designed a two-phase experiment which utilizes long-read nanopore sequencing in order to characterize and identify a diverse set of intergenerational epigenetic inheritance patterns associated with factors such as genotype, sex, parent of origin, and more. In the first phase of this experiment, we performed matched ONT and RNA sequencing on two inbred mouse strains and their heterozygous F1 crosses. We utilized this data to identify and map this diverse set of epigenetic inheritance patterns and then to further assess their effects on gene expression. In the second phase of the experiment, we wanted to go back and further characterize and validate a subset of these epigenetic inheritance patterns in the F2 generation. This F2 generation is made by crossing members of the F1 generation, and by virtue of the heterozygosity of their F1 parents, the genomes of these F2s will undergo meiotic recombination and independent assortment of chromosomes, which will enable this additional layer of characterization and validation. So as a direct consequence of this experimental design, the analysis of this data will necessitate an interstrain and allele-specific methylation comparison. In order to properly perform this comparison, we designed our own novel computational pipeline, loosely based around the traditional WhatsApp phasing approach. Our main goal with the design of this pipeline was to avoid the introduction of reference biases, which our lab, as well as others, have shown can be incredibly detrimental to these sorts of interstrain and allele-specific methylation comparisons. As such, our pipeline performs alignment and phasing relative to both reference genomes and extracts only those consensus phasing decisions. So effectively, those reads where allelic assignment is consistent, independent of the choice of reference genome. Furthermore, we've designed a novel tool which creates what we are calling an intermediate pseudo-hybrid genome, which is effectively a single unified coordinate system that represents all of the genetic diversity of our two reference genomes. And we perform all of our methylation comparisons within that unified coordinate system. So using this approach, we identified 10 distinct intergenerational epigenetic inheritance patterns. For the sake of time today, I'm not going to be able to go through all of those. So I'm just going to be focusing on the MEQTLs, or methylation quantitative trait loci. MEQTLs are genetic variants which are capable of influencing DNA methylation at specific CpG sites. There are many distinct types of MEQTLs, but they can be broadly classified into two distinct categories: cis-acting and trans-acting MEQTLs. Cis-acting MEQTLs require a direct connection between the SNP and the CpG over which it controls methylation. As such, these MEQTLs can only act over very short genomic distances and can only influence methylation at CpG sites that reside on the same allele as the genetic variant. Within our data, this will manifest as genotype and allele-specific methylation patterns because the genetic variant and its associated methylation pattern will be co-inherited on each allele. Now, this is all in stark contrast to trans-acting MEQTLs, which do not require this direct connection between the SNP and the CpGs of the MEQTL. As such, trans-acting MEQTLs can act over very large genomic distances and can influence methylation at CpG sites on either allele. Now, this introduces a trans-allelic competition between the two genetic variants of the MEQTLs to establish a methylation pattern over the same CpG sites, which further introduces the concept of dominance into trans-acting MEQTLs, which is not present in cis-acting MEQTLs. Dominant trans-acting MEQTLs themselves are a little bit more difficult to reason with, so for the sake of time today, I'm not going to be describing those. I'll just be focusing on the non-dominant trans-acting MEQTLs, of which there are two main types: incomplete dominant and codominant. In both of these scenarios, we expect to see one highly methylated and one lowly methylated inbred parental strain. In the case of incomplete dominant, we expect to see an intermediate level of methylation that falls between the highly and lowly methylated parents on both genetic alleles of the heterozygous F1 crosses. In the case of codominant transacting MEQTLs, however, you expect that within each genetic allele of these F1 crosses, you expect to see two clear epialleles, one matching the highly methylated and one matching the lowly methylated inbred parental strains. At the sample level, however, both incomplete dominant and codominant transacting MEQTLs will manifest with this intermediate level of methylation on the two genetic alleles of the heterozygous F1 crosses. And so we can see these patterns represented in the examples shown. With the cis-acting MEQTL on the left, you can clearly see this genotype and allele-specific methylation pattern, again, because the genetic variant and its associated methylation pattern are being co-inherited on each allele. And in the transacting MEQTL, we can see the clear disparity between the methylation pattern of the two inbred parental strains, and then we see that the two genetic alleles of the heterozygous F1 crosses exhibit this intermediate level of methylation, which falls between the highly and lowly methylated parental strains. So now to actually start distinguishing the incomplete dominant and codominant transacting MEQTLs, we have to start looking at the actual read level methylation data, because again, they are identical at the sample level. So again, in each of these cases, we expect to see one highly methylated and one lowly methylated parental strain. And in the case of incomplete dominant, we expect that every sequenced read on each of the two genetic alleles of the heterozygous F1 crosses will exhibit this intermediate level of methylation, again, falling between the highly and lowly methylated parents. For the codominant transacting MEQTLs, you again expect to see one highly and one lowly methylated parental strain. However, within each genetic allele of the heterozygous F1 crosses, there are two clear epialleles, one matching the highly methylated parental strain and one matching the lowly methylated parental strain. So now that we've identified and mapped these diverse set of epigenetic inheritance patterns, we wanted to go back and actually further characterize and validate a subset of them within this F2 generation. Now, as I mentioned earlier, these F2s are made by crossing members of the F1 generation, and during gametogenesis within these heterozygous F1s, meiotic recombination and independent assortment of chromosomes will enable the creation of an F2 population which is genetically diverse and also genetically distinct from their F1 parents. Now, this meiotic recombination and independent assortment of chromosomes effectively serves to separate distal loci within the genome. For cis-acting MEQTLs, which again can only act over incredibly short genomic distances, it's highly unlikely that recombination will occur between the SNP and the CPGs of the MEQTL. As such, if we perform targeted ONT sequencing of this DMR, the local genetics surrounding the DMR will reveal the identity of the genetic variant and as such be directly associated with the methylation level over the DMR itself. For transacting MEQTLs, which again can act over very large genomic distances, it's actually incredibly likely that meiotic recombination or independent assortment of chromosomes does occur between the SNP and the CPGs of the DMR. And as such, if we perform targeted sequencing of these DMRs using ONT adaptive sampling, the local genetics surrounding the DMR will give no additional information regarding the identity of that genetic variant, and as such will have no relation to the methylation level of the DMR. And so we can see these represented in these examples here. Sorry, there's supposed to be a little pink highlighting the DMR. So in the top left, we can see the cis-acting MEQTL in the inbred and F1 generation. We again see this genotype and allele-specific methylation pattern, and we see effectively the exact same pattern represented in the F2 population as well, indicating that recombination did not occur between the SNP and the CPGs of this MEQTL, and further solidifying this as a cis-acting regulatory mechanism. For the transacting MEQTL, we can see this aforementioned pattern where we have the divergence in methylation between the two parental strains and the intermediate level of methylation of both alleles of the heterozygous F1 crosses. Whereas in the F2 population, there's no clear relationship between the local genetics and the methylation of the DMR itself. So that's all I have for today. I think for the sake of time, I'm just going to speed through this conclusion slide and just say that nanopore sequencing, with its long reads, combined genetic and epigenetic information, as well as the simplicity and versatility of adaptive sampling, has really enabled a analysis of this epigenetic inheritance that was heretofore not possible. So I just want to thank my advisor, Andy Feinberg, as well as our incredible collaborators in the Threadgill, Hansen, and Macmillan lab. And thank you all for coming.